Hello, everybody, and welcome to our Precisionary webinar and scientific speaker series. Today, we're really honored to host Dr. Hanan Osman-Ponchet, who is the founder and CEO of PKDERM, which is a leading French biotech company specializing in innovative in vitro solutions for assessing the efficacy and safety of pharmaceutical and cosmetic products. And she has over 20 years of experience in drug development at Sanofi and Galderma, two large corporate giants. So this webinar is going to explore the occurrence of diffuse alveolar damage. So she's going to be talking about PCLS. And during this webinar, Dr. Osman-Ponchet will talk about the successful preparation of human precision cut lung slices, or PCLS, from surgical lung resections. And she uses our compressed-tone vibratome for her work, for her research. And we are going to be very excited to hear from you. So with that, Dr. Osman-Ponchet, I'm going to hand it over to you. Thank you so much for being our speaker today.
So thank you very much, Abby, for your invitation to do this webinar. I'm very happy to be there, and I hope that this webinar can be helpful for the audience. So here is a summary of the webinar. I will start with a short introduction about lung diseases, and I will focus on lung fibrosis. Then I will describe shortly the in vitro models that can be used to evaluate lung diseases and lung fibrosis. And finally, I will show you some case studies. So to start, as you know, the lungs are a vital part of the body, as they are responsible for breathing, circulation, and blood flow. However, the lungs are also susceptible to a variability of diseases. As you can see, the lung is divided into parts, the right lung and the left lung, and each lung has 3 lobes. Airway disease such as asthma and COPD make it difficult to breathe. And lung circulation diseases such as pulmonary hypertension and pulmonary edema can damage the blood vessel in the lungs and make it difficult for the heart to pump blood. Lung disease such as pulmonary fibrosis and sarcoidosis can damage the lung tissue itself and make it difficult for the lung to function properly. The most common symptoms of lung diseases are shortness of breath, wheezing, cough, chest pain, or respiratory functions. So, as I said at the beginning, I'm going to focus my presentation on pulmonary fibrosis. So pulmonary fibrosis, as you know, it's a chronic and progressive lung disease characterized by an excessive deposition of extracellular matrix protein in the lungs. This leads to a scarring and thickening of lung tissues and loss of lung function, making it difficult for oxygen to pass through. The exact mechanism of pulmonary fibrosis is often unknown, but it is believed to be a result of inflammation in the lungs. Actually, there is no really treatment for lung fibrosis. There is only two FDA-approved drugs to treat idiopathic pulmonary fibrosis, like pirfenidone and nintedanib. Both treatment have been shown to slow the progression of pulmonary fibrosis, but they cannot stop the disease. And also, we know that there is some side effect regarding this treatment. This is why we need to develop new treatment to treat the pulmonary fibrosis. Now, what about the pulmonary fibrosis and COVID-19? The data actually shows that about 35% of COVID-19 survivors appear to have developed pulmonary fibrosis. We call this post-COVID-19 pulmonary fibrosis. That is one of the most worrying pulmonary complications, as it causes permanent lung damage. Actually, we don't know the cellular and molecular mechanism for this complication, and the pathogenesis of this emergent syndrome urgently needs to be studied. So there is a huge amount of people who have pulmonary fibrosis. We showed that there is no really treatment to stop or to prevent this disease. This is why we need to work and to discover new treatments for. So what biological systems can we use to model this disease? So we can use 2D cell models, 3D lung models, or ex vivo models. Here I'm going to show you what we use actually at PKDERM to model pulmonary fibrosis. So we can use primary human lung fibroblast and also calcium-free human lung cell line. We also use a 3D lung model such as micellar from epithelix or epi-airway from MATEC. So this 3D model is very helpful because it contains the main cell type in the lung, like ciliated cell, goblet cells, and basal cells, and the tissue is cultivated at the air-liquid interface. The other model that I'm going to show you how we can prepare is the precision cut lung slices. So this model is helpful and it presents today the gold standard as it preserves the native architecture of the lung tissue. So how we can study pulmonary fibrosis in vitro? For that, the interest to use in vitro model, it can help to understand the mechanism of lung fibrosis and also help to develop new therapeutic strategies. In vitro model can be used to investigate the effect of pro-fibrotic stimuli, to assess the efficacy of potential antifibrotic agents, to to study the mechanism of lung fibrosis and also to assess cell migration in lung fibrosis. All the four features can help identifying lung fibrosis and explore the mechanism of this disease. And how we can assess the therapeutic potential of antifibrotic agents. For that, we need to use a multidimensional approach that use cell migration, gene expression, cell viability, immunofluorescence, proteomics, histology, and so on. Now I'm going to show you the first case study of cell migration and extracellular matrix markers using 2D model. In this example, the objective was to evaluate the efficacy of new drugs for the treatment of lung fibrosis. So before evaluating new drugs, we need to validate the model with well-known compounds. And the rationale behind that is that lung fibrosis is characterized by proliferation of fibroblasts with excessive accumulation of extracellular matrix within the lung. So targeting fibroblast proliferation and associated extramatrix deposition represent a logical therapeutic strategy for both delaying disease onset and also reversal of disease pathology. So for that, we use a primary human lung fibroblast and we stimulate with TGF-beta. And we treat with nintedanib or pirfenidone, the two FDA-approved drugs for lung fibrosis. In this study, we use Taxol as positive control. And we monitor cell migration using live cell imaging. And also, we measure the expression level of fibrosis markers using RT-qPCR. And here you can see we have several markers of lung fibrosis. So in practically how we can do, the lung fibroblasts are seeded in EBD insert to make a reproducible scratch. And then we treat the cell after 24 hours, and we use the Cytosmart Omni device to monitor at different time level the cell migration. And at the end, we make the analysis. So here you can see, for example, an example of how fibroblast can migrate in order to close the free cell gap. And at the end, we measure the kinetic or the rate of migration, and you can compare each product with the control. So here you have an example of the result can we get. We can see here that compared to the control, the untreated control, we can see that treatment with pirfenidone had no effect on cell migration. However, nintedanib markedly decrease cell migration in human lung fibroblast, and this is in accordance with the effect of nintedanib on cell proliferation of fibroblasts. So this is one approach of how to evaluate the mechanism of lung fibrosis. So the first and the most simple, let's say, approach is to look at the cell migration. Now, when we look at the expression of fibrosis markers, we can see when we treat the fibroblast with TGF-beta, we can see that we have an increase of all the lung fibrosis markers. You can see, for example, for FN1, fibronectin, ACTA2 for the actin, MMP12, COL1A1 and 3A1. And the most important increase we observed is with the LRRC15. This is a new marker today. We know that is involved in lung fibrosis. When we look at the effect of nintedanib on fibrosis markers, we can see that nintedanib decrease the expression level of FN1 and COL1A1, indicating the efficacy of the treatment with nintedanib on fibrosis marker. So the take-home message from this case study is that the primary culture of 2D human lung fibroblast offer a simple and cost-effective approach to modeling lung fibrosis. And this versatile model can be used in the early stage of drug development to evaluate the potential therapeutical strategies. The second case study I'm going to show you is about inflammation. And this example using the 3D lung model. The objective is to evaluate the efficacy of lung fibrosis treatment by targeting inflammatory pathway. And the rationale behind this is that, as I said before, the exact cause of pulmonary fibrosis is often unknown, but it is believed to be a result of inflammation in the lungs. So targeting lung inflammation represents a therapeutic strategy for delaying disease onset. In this example, we used the epi-airway 3D lung model, and we stimulate with LPS in order to induce the inflammatory markers, and we treated with beclospin in English, I think, using a nebulizer system. And we measure the expression level of inflammatory markers, IL-8 and IL-6. So what is the nebulizer system? In fact, the inhaler is a device used to deliver medication directly into the lungs. So this image shows a nebulizer system, which is a type of inhaler that delivers medication to the lungs in the form of fine and aerosolized particles. And of course, the size of the particles produced by the atomizer head determine how deep into the lung the particles of the medication will reach. So in our lab, we adapted this system to use an in vitro model. So here you can see how we adapted the system. In fact, we have the compressor. Here you have the nebulizer chamber. We can see here, and we sorry, we adapt the system to expose the tissue in the culture plate. And you can see we can conduct three experiment in parallel at the same time. So here what we can see using the Beclo-spin, you can see that when we treat the 3D lung model with LPS, we have increase of IL-8, but not increase of IL-6. And when the use of the nebulizer system with Beclo-spin to expose epi-airway tissues result in decrease in mRNA expression of both IL-8 and IL-6. So this example clearly shows that inflammation plays a pivotal role in the initiation and progression of pulmonary fibrosis, and 3D lung tissue models and the inhalation route provide a powerful tool for studying inflammation induction and its implication for lung fibrosis. Now we saw the 2D model that is very simple and cost effective. We saw a more complex model, the 3D lung model. And now I'm going to show you another 3D model, because it is also a 3D model, the model of PCLS. And this case study is focused on a profibrotic cocktail and how to induce fibrosis in this model. So, what are the human precision cut lung slices, or hPCLS? So hPCLS are thin sections of lung tissue that are precisely cut using a microtome. They retain the native architecture and cellular composition of the lung, including the airways, blood vessel, and immune cells. The advantage of hPCLS are the preservation of lung architecture, responsiveness to stimuli, and suitability for drug screening. This model can be applied to study lung fibrosis, to investigate respiratory infection, and also in modeling lung toxicology. Now, how to prepare hPCLS? Simply speaking, I show you here the different steps to obtain finally the PCLS. The first step is to obtain lung resection from surgery. Then the second step and the more challenging step is the inflation of the lung with low melting agarose. Here you can see the lung after inflation with agarose. After that, we perform a biopsy punch of 8 millimeter of diameter. You can see here the lung, the punch. Then the punch of lung is mounted on the holder, and we put also the agarose in the holder in order to maintain stably the punch. And we use this device, it is a cold device, in order to gellify the agarose and to make rigid the system. And then we prepare the vibrating microtome, the compressed-tome VF300OZ. So we put the holder with the biopsy of lung in this compartment of the system. You can see here the punch of the lung. We fill the container with a cold buffer. And here you can see we have the first PCLS that we can get. You can see here the image of PCLS. And here you can see how exactly it works. This is a very short video to see. It is done automatically. So we can cut different slices. And the system is calibrated to have a slice of 300 micrometer with 8 millimeter of diameter. So you can see the slice with the agarose around them. So so far in our lab, we had 9 lung resections from 9 donors. Each the lung resection had between 0.8 and 7 grams. And we obtained per preparation between no slice, no PCLS, 0 PCLS to 200 PCLS. So you can see that this is very variable according to the quality of the lung, to the shape, also the thickness of the lung, and sometimes the inflation of agarose is very hard to do, very difficult. So this is a very challenging step. So at the end, the PCLS are cultivated in 12 or 24 well plate in cell incubator. In terms of characterization of PCLS, we can monitor cell viability, and also we can monitor the actin disposition. We can monitor gene expression. We can do a lot of things with this model. So here you have the results of cell viability of 3 donors, and you can see that the viability of PCLS is increasing with the duration of culture. We did the experiment between day 2 and day 14 after preparation, and at day 14 for the 3 donors you can see the viability increase, and this is well documented in the literature as the PCLS. There is a recuperation, compensation after the preparation. So in this example, the viability was measured using the entity assay. We can use another assay to measure the cell viability, is the live dead assay using the microscopy. And you can see on this image, we can really observe the architecture that is well preserved in the different PCLS from different donors, and the red color is dead cells, and the green color is live cells. And when we measure, we have majority of the color is green, meaning viability is well preserved at day 7 in this experiment. So this is an example of what we can do regarding this model. Now, how we can induce lung fibrosis using the PCLS. Here you have the composition of the profibrotic cocktail, and this we use this cocktail as indicated in the publication of Al-Safadi of 2017. So we treat the PCLS for 48 hours with the profibrotic cocktail, and then we add the nintedanib and the profibrotic cocktail for another 72 hours. And at the end of the treatment, we do the analysis of different endpoints. So here I'm going to show you the results of mRNA expression of some fibrotic markers in human PCLS on 3 different donors. So we can see that the treatment with the profibrotic cocktail in blue increase increase the expression of some fibrotic markers, mainly the FN1 and the COL1A1. And also we can see that the treatment with nintedanib can reverse this effect. Here you can see in gray in some donors. And also we notice that there is a high interindividual variability that can be observed between the donor. For example, for donor 5 and donor 6, we don't have the same effect of the profibrotic cocktail regarding the ACTA2, for example. In donor 5, we don't have any effect. However, in donor 6, you have an increase of the expression of this gene. And this is normal. Why? Because we know that according to the donor, according to the historical medication, we expect such variability. This is why when we do this kind of studies, we need to do more than 3 donors in order to get an average of the results when we evaluate a new treatment. Here you have an example of the staining of actin in PCLS treated with the profibrotic cocktail and with nintedanib in 2 donors, and we can see, in fact, we cannot see actually any effect regarding the actin staining. We do the same thing on another donor, the donor 7, and we confirm that this endpoint is not useful to identify any effect of the treatment on actin. And what we are going to do now is to make histopathology analysis in order to see more deeply if there is any changes in the architectures of the PCLS. So and also what we are doing now in order to ameliorate and to improve the model, we are trying to cultivate the PCLS at the Earth liquid interface, because some evidence, some data shows that it is the results may be better, and the model is more close to the real life when the PCLS are cultivated at the interface air liquid. So the take-home message from this example is that the preparation of human PCLS is a complex and technically challenging process that requires expertise in tissue handling and culture technique. This is due to the delicate nature of lung tissue and the need to preserve its native architecture and the cellular composition. However, when the human PCLS preparation process is mastered, it provides a valuable tool for studying lung fibrosis due to its ability to recapitulate in vivo environment and respond to various stimuli. So this is why this makes human PCLS a valuable tool for investigating the mechanisms of lung fibrosis, evaluating the efficacy of potential antifibrotic therapies, and developing therapeutic strategies. And to summarize this webinar, I showed you that in vitro models have become a valuable tool for assessing the efficacy of treatments for pulmonary fibrosis and also other lung diseases. While each model has, of course, its advantages and limitations, the key to success research lies in selecting the most appropriate model for the research question at hand. And the PCLS model is currently considered the most relevant model for studying lung disease, as it preserves the architecture and of cell types of the lungs. And I would like to thank you very much for your attention, for attending this webinar, and I'm open to your questions. And I would like just to thank my team, Laurie Perdigon, Manon Barthes, and Jean-Paul Thénault. And please ask your question.
Thank you so much, Dr. Osman-Ponchet. We actually had a comment from Dr. Pineda, who is one of our really first and foremost compressed-dome PCLS experts. So Dr. Pineda, do you want to unmute yourself and comment or have any questions? Oh, it says his microphone is not working. So I'll ask it on say it on behalf of you. It says he wants to thank you for very nice work. He would recommend moving away from the FITC channel due to the lungs' high autofluorescence. So maybe Dr. Osman-Ponchet, can you comment a little bit about the autofluorescence and imaging in the lung and how it might impact your interpretation of the results?
Sorry, could you please repeat the question because I didn't understand the beginning of the question.
Yeah. So Dr. Pineda made the comment that he would recommend to move away from the FITC channel due to the lungs' high autofluorescence. So maybe you can share with the group your thoughts about autofluorescence and imaging in lung slices.
Okay. In fact, we didn't see autofluorescence in our case because what we do here, in fact, we take the PCLS and we stain with with the phalloidine conjugated to FETC. So the PCLS that not stained, it doesn't show any fluorescence in our case. But this is a good advice. We will try to use another channel than FETC, and we'll see if we saw something better.
Wonderful. Thank you. And I want to open up the room here to see if anybody has any questions. Please feel free to unmute and ask them or enter them into the chat box.
There is something in the discussion.
Yeah, just thank you to comment on a great presentation. If there are no further questions, our webinar is recorded. So again, and feel free to reach out to us at Precisionary and me directly if you wanted to have additional questions or follow up. So Dr. Osman-Ponchet, thank you so much for your talk on PCLS and your work and looking at lung fibrosis. We're really honored to support your research and to host you. And with that, thank you all for the attendees as well. See you soon.
Yeah, thank you very much. Bye-bye.
Bye-bye.